Hello everyone, before we proceed with the video, I would like to give credits to the creator of the slideshow, which are the UNIMAS lecturers that teach biochemistry. And now we will proceed with the video, in which I will be explaining about the basics of glycolysis, Krebs cycle, and electron transport system, or simply say ETS. And I will proceed with glycolysis. Glycolysis is a glucose catabolism that is carried out in all cells. What happens to the glucose molecules? In the glycolysis process, the glucose is converted into pyruvate, and three final products are formed, which are two pyruvate molecules, 2 ATP and 2 NADH. After the glycolysis process, there are three possible phases for pyruvate. It may be undergoing aerobic oxidation, anaerobic glycolysis, or anaerobic fermentation depending on the abundance of oxygen. There are two phases in glycolysis. The phase 1 would be the preparative phase, in which the 6-carbon sugar or 6C sugar is converted into a 3C sugar, and the phase 2 ATP generating phase, which ATP will start to phase off in this process. And there's a net gain of 2 ATP per glucose molecules consumed. As the glucose is oxidized, 2 NAD plus are reduced to 2 NADH, as in the NAD plus ions, one hydrogen ion is accepted for each and converted and reduced into 2 NADH molecules. If the oxygen is in abundance, aerobic oxidation would be carried out by using the pyruvate and NADH produced earlier, and if oxygen is not sufficient, anaerobic glycolysis and anaerobic alcoholic fermentation would be carried out in the further processes. In anaerobic conditions, the first one would be lactic acid fermentation, which usually occurs in our muscle cells if we are undergoing high density activities such as sprinting, jogging, swimming, and other extreme sports, and any other activities that use high muscle capacity. Alcoholic fermentation usually occurs in yeast, and it's alcoholic, so it's basically a fermentation process that occurs in wine making or beer production by using yeast, and yeast usually undergoes anaerobic respiration, which will in turn produces carbon dioxide that fermentates the alcohol. And when oxygen is present, most organisms will undergo two more steps, which I will be further explaining later. The first one will be Krebs cycle or citric acid cycle, and the second one will be electron transport system or ETS to produce more ATP. Compared to anaerobic respiration, aerobic respiration produces significantly more ATP because oxygen is present and more products would be formed. So we'll proceed with Krebs cycle or citric acid cycle. Pyruvate, the first products that in turn being substrate for the next reaction. Pyruvate is the product of glycolysis that we had discussed a moment ago. It first altered in a transition reaction by remodeling of a carbon and two oxygens which form carbon dioxide. And basically, Krebs cycle is a process where the product formed will be a substrate for the next reaction, and it will go on and on. Well, please take a look at the notes attached here. Basically, Krebs cycle is a cyclic process that oxaloacetate reacts with acetyl CoA to form citrate to start a series of several other reactions. And these are the further processes of Krebs cycle, and some of the key processes that I will be highlighting here is the process. Actually, everything is important, but the most significant one would be the conversion of alpha ketoglutarate acid to succinyl CoA, which are the processes labeled 3 and 4, and the processes between 4th and 5th, which you can see an ATP there, is a conversion of ADP and a phosphate ion to ATP. These are the significant processes in Krebs cycle that processes ATP. and also at the end of the process, ATP would be produced. And again, please take a look at the notes attached here. And something worthy to mention that there's a few sub-products and intermediate products formed in Krebs cycle, namely NADH and FADH, and also FADH2. FADH2 is not as rich an energy carrier as NADH because they yield less ATP compared to NADH. The carbon dioxide released by cells is generated by the Krebs cycles. Like in our respiration process, well, I think all of us here should be known that we take in oxygen and we breathe out carbon dioxide. So the carbon dioxide released by cells is generated by the Krebs cycle, as are the energy carriers, which are NADH and FADH2, which plays a role in the electron transport system that we will be proceeding later. Basically, Krebs cycle converts pyruvate to carbon dioxide, NADH, FADH2, and also GTP or ATP. So please take a look at the overall Krebs cycle reaction. This reaction is basically what happens in the Krebs cycle. It summarizes the whole process. The reduced energy is used to generate ATP using the electron transport chain in the presence of oxygen. So when oxygen are present, well, as I mentioned earlier just now, aerobic respiration would be used instead of anaerobic respiration. And in aerobic respiration, since there are oxygen, reduced energy is used to generate ATP, and subsequently, more ATP is generated. Oxygen is basically like, how would I explain it? It's an additional sum of, it's basically like a sum of money that helps in a business. So when we got more models in our hands, we will spend less and subsequently we will earn more. It's the same with the relation of oxygen to ATP. Subsequently, 36 ATP are produced in aerobic respiration compared to only 2 produced in anaerobic respiration. And now, before we end this video, there's one last segment in electron transport system or ETS. While Krebs cycle occurs in the matrix of mitochondria, ETS chemicals are embedded in the membranes known as the cristae. It's basically the second membrane in the mitochondrial cell. Mitochondrial organelle, sorry. Krebs cycle completely oxidizes the carbon in pyruvate, producing a small amount of ATP and reducing NAD and FAD into higher energy forms. And in the ETS, this NADH and FADH2 is used to produce ATP in structures called cytochromes. Oxidative phosphorylation is the gradient exchange of H+ ions across the inner mitochondrial membrane, and this mechanism is also known as chemosmotic coupling. This involves both chemical and transport process, and this will cause a drop in the potential energy of electrons moving down the ETS chain occurs at 3 points. At all these 3 points, the ATP are converted into ATP, and the potential energy is captured by ATP and stored in the phosphate bond, pyrophosphate bond. And well, please take a note. Please go through the notes here. The energy formed from the transfer of electrons along the chain transport protons across the membrane and creates an electrochemical gradient. Hence the name electron transport system. So the energy from the transfer of electrons, the energies go through from the chain transport protons across the membrane from one membrane to another. This creates a difference in the potential or electrochemical gradient, and this would be captured and transferred and converted into the form of energy. As the accumulating protons follow the electrochemical gradient back across the membrane through ATP synthase complex, the movement of the protons provide energy for synthesizing more and more ATP from ADP and phosphate. the end of this ETS, 2 protons, 2 electrons, and half of an oxygen molecule or a single oxygen molecule combine to form water. Since oxygen is the final electron acceptor, this process is called aerobic respiration. Also, please recap what I had mentioned earlier. If oxygen is present, this process will be ended by oxygen as the final electron acceptor, and at the end more ATP would be produced. And this under summarization of aerobic and a comparison of aerobic and anaerobic respiration and the amount of ATP produced. Anaerobic fermentation results in 2 ATP per glucose molecule, and again, anaerobic fermentation is a process where usually occurs in muscle cell when we are undergoing an high intensity activities, or in the yeast cells, as yeast usually carries out anaerobic respiration or fermentation in the wine producing process. While oxidative phosphorylation basically refers to aerobic respiration. They can yield up to 38 ATP per glucose, and the rate of ATP production via anaerobic glycolysis can be 100 times faster than that of oxidative phosphorylation. So, when we are undergoing a high intensity activity, anaerobic respiration would instead replaces the aerobic respiration in the muscle cells, because the muscle cell needs lots of ATP in a short amount of time. And the overall energy balance per glucose molecule is basically divided into the investment phase, which are the same for aerobic and anaerobic respirations. 2 ATPs are used in glycolysis. 2 ATP, in other words, 2 ATP are invested before the payoff phase, which we obtain the returns from the investment of 2 ATP. In aerobic respiration, there are lots and lots of ATP formed from the glycolysis and subsequently from the Kreb cycle and ETS, electron transport system, and results in a total of 38 ATP, while in anaerobic respiration, there's only 2 net gain of ATP produced. And with that, I'll end my video on explaining the basics of glycolysis, Kreb cycle or citric acid cycle, and ETS. And thanks for watching.